Good morning. Uh, we will acquire today some introductory ideas on proteins. This lecture is under the course Protein Technology, course code OEFT 602B. My name is A.K. Mukherjee. I am Assistant Professor in the Department of Food Technology, Haldia Institute of Technology. The word protein is derived from Greek word proteos, which means primary or of prime importance. Definitely, proteins are of primary importance, prime importance, because they are responsible for cellular growth and repair process within the cell, starting from lower microorganism and up to higher animals. Proteins are composed of long chains of amino acids joined together via peptide bond. That is, proteins are polymers. They are macromolecules. They have very high molecular weight and they are polymers of amino acids. Once a protein is hydrolyzed by using strong acid, concentrated hydrochloric or sulfuric mineral acids, the amino acids are obtained. These amino acids are different amino acids. Commonly, they are 20 different amino acids in different numbers present within the protein. One recently, another amino acid is found in different enzymes, which is selenocysteine, that makes 21 different amino acids which are found naturally in different proteins. However, a large number of amino acids are there which are discovered so far, above 300 their numbers. Only 20 to 21 are common. So, proteins are heteropolymers. Once the protein is hydrolyzed, not a single type of amino acid is obtained, but different types of amino acids are obtained in different number. A polypeptide with more than 100 amino acid residues having molecular mass more than 10,000 unit is called a protein. So proteins are having very high molecular weight. Amino acids are derivatives of carboxyl. Now let us learn what is amino acid. We can see the structure here, amino acid. Carbon, carboxylic group is attached to a carbon. That means it is the alpha carbon. If we consider this one as the main chain, the functional group is carboxylic acid, and that is why the compound is called amino acid. Amino group is the side chain. However, this structure is not available in aqueous form. When protein is within a solution, water, this structure is not available. Protein are always having some polarity. Amino acids having some polarity, and they will have some charges either on the nitrogen or on the carboxylic group. They are derivatives of carboxylic acids formed by substitution of alpha hydrogen. This carbon is called alpha carbon, just adjacent to the functional group, and with this alpha carbon this hydrogen is attached, that is why it is called alpha hydrogen. Amino acids are alpha amino acids because this amino group is also attached to this carbon. Amino acids are either D or L. Naturally, amino acids are L amino acids because this carbon is chiral. As long as this R group is different from these three groups, this carbon is chiral and will give two optical isomers, 2 to the power 1, that means two isomers. One is dextrorotatory, another is levorotatory. Natural amino acids are all levorotatory, except glycine. Glycine having this R group as hydrogen, So there is no chiral carbon. That is why it is not optically active. Amino acids are optically active molecules due to the presence of a chiral carbon. This is shown in the three-dimensional structure. According to new IUPAC nomenclature, LD forms were replaced for S and R systems. Proteins play key roles in living system. Different roles they play. Grossly, proteins can be classified as structural protein, proteins of cell available naturally within the cell, structural protein, and functional protein. Structural protein are fibrous protein. They are insoluble in water. Collagen, elastin, alpha keratin, etc. Those are structural protein. They give rigidity and structural property to the system, and functional property. Those are actually globular protein. They are soluble in different solvents, and if these are enzymes, they catalyze the reaction within the cell. If this is other globular protein, it has other functions. They play some functions. Immunoglobulin, hormones also, they are proteins. Transport protein, hemoglobin transports oxygen. That is also a protein. Information transfer. Hormones transfer information to the different parts and to the central nervous system of the body. Basic unit of protein is amino acid. Amino acid forms deuterion, as well as protein also forms this deuterion structure. What is a deuterion? Since the amino acid having one amino group and another carboxylic group, if we start adding alkali to a highly acidic solution of amino acid, which is at a lower pH initially, the carboxylic group is undissociated under that acidic condition, while the amino group, the lone pair of electrons on the nitrogen, will have one proton and it will be NH3+. Now we are adding alkali slowly and slowly, and the carboxylic group will be dissociated and will form carboxylate anion. At a certain pH, a structure of protein will be available where both amino and carboxylic groups are ionized. The amino group is ionized as positive charge and carboxylic group will carry a negative charge. They will interact, that is intramolecular attraction, and they will make the molecule electrically neutral. This structure is called deuterion structure, and this structure is available only at a certain pH where both these amino and carboxylic group will carry opposite charges, and that is called isoelectric pH. At isoelectric pH, amino acid is electrically neutral and amino acids get precipitated. Proteins also behave in this manner because protein is a linear chain of amino acids, peptide bond. Once they are unfolded from their tertiary and secondary structure, the terminal two groups, one is amino group, another is carboxylic group. If we start from amino side, then the first amino acid will have carboxylic group on the other side. This carboxylic group will bind the amino group of the second amino acid, and so on. Finally, the last amino acid will have its carboxylic group free and will act similarly like a single amino acid to form this deuterium structure. 20 common amino acids. Glycine, alanine, these amino acids are actually the non-polar amino acid. Glycine, alanine, valine, these amino acids. Leucine, asparagine, tryptophan, glutamine. Some are acidic amino acid, aspartic acid, glutamic acid. Some are basic amino acid, some are neutral amino acid. Proteins are linear polymers of amino acids, as we have already discussed this thing. So, by condensation, this peptide bond is formed. Now, once ammonia react, this is the water molecule is removed and ammonia is reacting, amino group is reacting and forming this peptide bond. Classification of protein. Globular protein. Proteins that consist of long chain of amino acids folded up into complex shapes. Globular proteins serve as or form important components of the following: enzyme, hormone, antibodies. Antibodies are also protein in nature. They recognize different foreign invaders, antigen, and they bind those antigen and make them inactive. Some structural proteins, as we have already discussed, fibrous protein, those are also there. This is structure of insulin. There are two peptides actually in insulin. One is in this bent form and sulfur bridge is there between these two peptides. And this is the first such peptide, A chain and B chain of insulin. Insulin is responsible for uh, limiting the concentration of glucose within the blood. Proteins that tend to be fibrous protein tend to be insoluble and strong and so play a structural role in organisms for support or protection. They can be subdivided into several different types. Keratins found in hair, fingernail, and bird feather. Collagen, the most abundant proteins in a vertebrate body, found in connective tissues such as cartilage. Elastin, found in ligaments around the blood vessels. So, structure of keratin, you can see here, this is the structure. Protein describe four different structure. First such structure, which is the peptide bond, a covalent bond is formed between amino acids, forming the primary structure of protein. It is called a polypeptide. A number of peptide bonds are available within the molecule, that is, it is called a polypeptide. So polypeptide is a linear chain of different amino acids. This polypeptide is bound further through hydrogen bond and it produces helix structure or a beta pleated sheet structure. Difference between globular and fibrous protein. They are more or less spherical in shape, globular protein, while the fibrous protein polypeptide chains which lie side by side held together by hydrogen bond and have thread-like structure. It is actually gives the rigidity, this type of arrangement, to the part of that particular cell which contains this fibrous protein. Globular proteins are usually soluble in water. Also, many globular proteins soluble in different non-aqueous solvent. Fibrous proteins are insoluble in water. Insulin, albumin, etc. And example already given, keratin, etc. Structure of protein. Each protein has a unique structure, amino acid sequence, different amino acids. So repeating unit are there. Once K is here, another K, same amino acid will be found here. The sequence is like that. However, this sequence we cannot dictate over. This is the natural sequence making a natural protein. Basic structure unit of protein. Primary structure, as we have said that it is composed of a peptide bond, a number of peptide bonds in a polypeptide. The sequence in which the amino acids are arranged in a polypeptide chain of a protein is called the primary structure of the protein. Now, secondary structure. These are two types of structural protein. Alpha helix, the peptide chain coils and turns in the form of a coil. It is like a coil like this or this, either right-handed or left-handed. However, right-handed helical structure is most common. Another type of secondary structure is called beta sheet or beta pleated sheet. This beta sheet joins two different polypeptide, either parallelly or anti-parallelly. That means if this is the amino terminal and this is the carboxylic terminal of a polypeptide, another polypeptide, if this is amino terminal and this is the carboxylic terminal, it binds in this manner. This is called the parallel binding. Another, if any one is reversed, it will be like this. This binding will be this. So amino will come here and carboxylic will come in the next polypeptide. It will bind like this. So this beta pleated sheet. So if both are present in a structure, it will be like this. So this part is actually beta pleated sheet. Here the protein chains are not coiled but straight join is there. Now once enzymatic reaction takes place, this portion, since many R groups etc., those are hidden within the coil. This coil is formed, alpha helix is formed through hydrogen bonding, and this hydrogen, a single hydrogen bond is very weak bond. However, large number of hydrogen bond together playing a role to make this structure very strong. Where this hydrogen bond is formed, the peptide bond of two successive amino acids, between two successive amino acids, one is a COO group. In a peptide, there is a COO group, and the COO group followed by NH group. So in a single peptide, this group is there. In the next such peptide, another such pair of groups will be available. So oxygen of CO binds through hydrogen bonding of hydrogen of NH. Now comes this secondary structure, alpha helix. Alpha helix C is the helical structure. However, this is a three-dimensional structure shown where many amino acids making this structure complex, and these are the hydrogen bonds. This is the helix. Secondary structure, alpha helix and beta sheet. Beta sheet is there. This is alpha helix and this is the beta sheet portion. So this is very straight chain. When enzymatic reaction takes place, enzyme first of all finds its way to come in contact on the beta sheet and making two polypeptide, if joined together through beta sheet, separate. This is the beta sheet structure. Secondary structure, beta sheet have regular hydrogen bonding pattern. Another diagram, it is very complex if it is drawn, otherwise it could be simpler for understanding. Tertiary structure. Secondary structure is made of hydrogen bond. There is no covalent bond acting to develop the secondary structure, either the alpha helix or the beta sheet. However, in tertiary structure, a type of covalent bond which is called salt bridge or sulfur-sulfur bond, disulfide linkage, that is available, making the structure stronger than any other structure, similar to peptide linkage. Sulfur, if sulfur containing amino acids are there, disulfide linkages are found. So further coiling of the protein takes place. What happens is, here if I say this is the helical structure, it will again coil like this due to this sulfur-sulfur linkage. And so further coiling and making it more complex, the structure being more complex, composed of two or more polypeptide chains. In case of quaternary structure, however, tertiary structure contains this sulfide-sulfide linkage. At the same time, the tertiary structure also has hydrogen bond, and also electrostatic force of attraction, because a cluster, a part of a polypeptide, if it develops a positive charge, another part develops a negative charge, there is further attraction making the coil further folded, and it is like a very complex structure in tertiary structure. Weak van der Waals forces are also available in tertiary structure. Quaternary structure is not available in all types of protein. It is not so common, but it is found in some protein like hemoglobin. Hemoglobin consists of two polypeptide chains, and in between two polypeptide chains, this further electrostatic attraction, etc., they make this structure coiled, and two or more polypeptide chains join to form this quaternary structure. This is the protein structure. Three-dimensional structure of different proteins. This is tertiary structure, and this is a quaternary. Quaternary is different color, shows different polypeptide chains which are joined through different weak forces, not covalent binding. Amino acid sequence, primary structure, secondary alpha helix beta sheet, tertiary structure, three-dimensional structure formed by assembly of secondary structure, quaternary structure formed by more than one polypeptide chain. Close relationship between protein structure and its function. Example of enzyme reaction. This enzyme reaction, two different hypotheses are there. One is lock and key, and another is this induced feed model by Postland. So these enzymes are protein in nature. However, enzymes also have a non-protein part that is called coenzyme or cofactor. It can be a metal element or it can be a vitamin like substances. So matching the shape to A, so a substrate just fits to a particular enzyme. That enzyme will catalyze the reaction of that particular substrate. It is called the lock and key model. And induced fit model is also there, which is more acceptable nowadays, which says once the substrate comes and binds the enzyme, there are conformational changes taking place, and that makes the enzyme to find an alternative path with lower activation energy to carry out the reaction. Hormone receptor, antibody, etc. Those are having this native state. At normal pH and temperature, each protein will take up a shape which is energetically most stable. The shape is called the native state of the protein. The protein in its native state, either soluble or insolvent, but once thermal treatment, acid or alkali, if it comes in contact of those things, heat, acid, alkali, these proteins are unfolded. That is, we call this process coagulation of protein or denaturation of protein, unfolding of tertiary structure, and exposing of R group. R group may carry polarity or it may be neutral depending upon the type of amino acid present in the R group and amino acids available within the protein. And suddenly the protein will change its physical property. That means solubility in solvent or water, and its attraction and separation from that solvent. All those things will change once those native structure is destroyed for that protein. Protein denaturation. Denatured state, unfolded state. Native state, folded state. Denaturation, heat, urea, salt, acid, alkali, etc. So, thank you. So, thank you for your kind attention. Today, we end this lecture.